Practical 5, Assays for Specific Metabolic Activities, Group 7, Subgroup 6. This is our group members for Subgroup 6.
For introduction, microorganisms have several central metabolic pathways involved in the flow of energy and carbon through the cell. Some heterotrophic bacteria can metabolize sugars or complex carbohydrates to produce energy. The ability of certain bacteria to utilize carbohydrate for energy source either by oxidative or fermentative metabolisms used to identify their species. Fermentation is the term applied to the anaerobic breakdown of carbohydrates or sugar. The end products of fermentation often vary from one organism to the other, depending on the pathways involved. There are also some bacteria that can use citrate as their only carbon source. The capability to utilize citrate is determined by inoculating the bacteria on Simon citrate agar. The bacteria will use the enzyme citrase to degrade the citrate and also convert the ammonium phosphate to ammonia and ammonium hydroxide.
In this slide, there are listed materials used in Practical 5. For Part A, acid and gas production from carbohydrate fermentation. We have been using tubes of sterile purple broth based with inverted Durham tubes, filtered sterilized solutions of carbohydrate, 10% of glucose, fructose, lactose, sucrose, and mannitol. We have been using pipettes. For Part B, Vogel's Prescure task. Firstly, we've been using sterile tubes of MRVP medium, Barrett's reagent A, Barrett's reagent B, and pipettes. For Part C, methyl red test, we have been using sterile tubes of MRVP medium with following a methyl red solution. Lastly, for Part D, citrate utilization, we have been using a slants of Simon citrate agar. So this is the methods for every part in one video.
These are the results and interpretation. For part A, acid and gas production from carbohydrate fermentation. In tube A, the microorganism used is E. coli. In tube B, the microorganism used is Enterobacter aerogenes. And these are the results for carbohydrate, acid production, and gas production. For part B, Vox-Proskauer test, for tube A, the microorganism used is E. coli. The medium color remain yellow. For tube B, the microorganism used is Enterobacter aerogenes. The medium changed to pinkish red polymer in the presence of acetone. For part C, methyl red test, in the A tube, the microorganism used is E. coli. An immediate red reaction signifies a mixed acid fermentation, which shows a positive result. In the B tube, the microorganism used is Enterobacter aerogenes. The yellow solution remain unchanged, indicate a negative result. For part D, citrate utilization, in the first lens of Simon's agar, A shows a green color which indicates a negative result. The microorganism used in tube A is E. coli. In the second slant of Simon's agar, B shows a royal blue color which indicates a positive result. Enterobacter aerogenes has been transferred in this tube.
Discussion: Acid and gas production from carbohydrate fermentation. Fermentation is a chemical process which molecules such as glucose are broken down anaerobically. When microorganisms ferment carbohydrate, an acid or acid with gas are produced. The final products can vary depending on the organisms and substrate being fermented. Lactic acid, formic acid, acetic acid, butyric acid, butyl alcohol, acetone, ethyl alcohol, carbon dioxide, and hydrogen are typical byproducts of bacterial fermentation. The pH indicator changes color to indicate that acid production has lowered the test medium's pH. Bacteria may use the peptone to make alkaline byproducts, thus color change only happens when enough acid is produced. Acid production turns the medium yellow as a result of organism fermenting the provided carbohydrate and producing organic acids, which lowers the medium's pH and make it acidic. Acid and gas production turns the medium yellow as the provided carbohydrate is fermented by the organism, producing organic acids and gas in the process. The appearance of tiny bubbles in the inverted Durham tubes indicates the presence of gas generation. Absence of fermentation, the red color of broth does not fade. The organism grows in the medium, utilizing other energy sources even though it is unable to use the carbohydrate. Based on the results of the experiment, it is shown that tube A, which contained Escherichia coli, fermented and produced acid and gas as the color of the broth changed to yellow and there is tiny bubbles in the inverted Durham tubes. This showed that Escherichia coli have aerogenic response and anaerobic bacteria. The result is same for tube B, which contain Enterobacter aerogenes, which determine that Enterobacter aerogenes have aerogenic response and anaerobic bacteria as fermentation occur. The Voges-Proskauer test is used to determine if an organism produces acetyl carbinol from glucose fermentation. Butanediol is one of the end product of fermentation pathway. This pathway produces acetyl-methylcarbinol, or in other name is acetone, which is neutral reacting end product. If present, alpha-naphtol, a strong alkali such as 40% KOH, and ambient oxygen transform acetyl-methylcarbinol into diacetyl. The components in the broth peptones that contain guanidine and diacetyl then condense to create a pinkish red polymer. Results is positive for tube B, which contain Enterobacter aerogenes, and negative result for tube A, which contain Escherichia coli. This showed that Enterobacter aerogenes produce acetone as the chief end product of fermentation, as the broth color changed from yellow to pinkish red polymer. Meanwhile, tube A, the color remained the same, which shows that Escherichia coli lack of complete butanediol fermentation pathway. Methyl red test. The methyl red test detects the production of sufficient acid during the fermentation. Certain bacteria can use glucose as an energy source and produce stable acids as a byproduct, such as formic, acetic, or lactic acid. The stable acid is produced by this bacteria through further metabolism of pyruvic acid, which is first converted from glucose by the mixed acid pathway. The kind of acid that is produced varies from species to species and is determined by particular enzymatic pathway that the bacteria have. Methyl red changes from yellow to red as a result of the acid generated, which lowers the pH to 4.5 or below. For every mole of fermented glucose, the mixed acid route yields 4 mole of acidic products, 1 mole of neutral fermentation products, 1 mole of CO2, 1 mole of H2. The pH of culture medium significantly drops as a result of high amount of acids generated. From the results, tube A, which contains Escherichia coli, have ability to utilize glucose with production of stable acid as the color methyl red change from yellow to red. Tube B, which contains Enterobacter aerogenes, the color of methyl red remain the same, indicate that there is no production of stable acid. Lastly, citrate utilization. Citrate agar is used to test an organism's ability to utilize citrate as a source of energy. Citrate agar contains citrate, and the only nitrogen source is inorganic ammonium salts. The enzyme citrate permease, which is produced by bacteria that can proliferate on this medium, can convert citrate to pyruvate. Pyruvate can then be used by the organisms to produce energy through its metabolic cycle. Growth indicates that the Krebs cycle's intermediate metabolite, citrate, is being used. The ammonium salts are converted to ammonia by the bacteria as they break down citrate, which raise alkalinity. Above pH 7.6, the medium's prometamol blue indicator changed from green to blue due to the pH shift. From the experiment, tube B, which contains Klebsiella pneumoniae, shows a positive result, and tube A, which contains Escherichia coli, shows negative result. Escherichia coli lacks a citrate transport and oxaloacetate decarboxylase, which is a reason why it can't use citrate as a sole carbon source.